So this week we are learning all about the functional components of cells, right? And all of the bits and pieces that make cells live, that allows them to live, right? To be something that we say, this has life. And one of the questions that may not have occurred to you, but it's this idea is that why are cells so small? Why are they so small? And why are we made up of billions and trillions of cells instead of just being one giant cell, right? So let's dive into that question because that explains a lot about how our chemistry functions and how cells interact with each other. And I want to start first by showing an animation of a realistic view, for the most part, of what it looks like to be inside our cells on a cellular protein molecular level. Okay, so let's switch over and watch that animation. So this video, we're not going to watch all of it. I'm going to kind of show bits and pieces. It was done by some scientists at Harvard, and it is not, I guess, imaginary. It's not fantasy. It's based on models that we have in science that we've done to understand how proteins are structured, how cells interact with each other, how everything works inside of a cell. Okay, so we're going to watch parts of this. I'm going to post another link to a video of a professor that I know that explains what's going on in each step, so maybe that would be helpful and will also show us really cool stuff about all the stuff that we've learned. So again, this is a realistic depiction of what it looks like inside of our cells and what's going on between our cells. Okay, so this section right here, so these are cytoskeleton components. Okay, so these are proteins, these are filaments that are, there it looks like that's an enzyme, yep. So that cleaved apart some of the subunits of the protein. So again, more filaments of the cytoskeleton, fibrous filaments in the cytoskeleton, just proteins, polymerizing and depolymerizing, okay? And we've seen this one before, I've showed this clip before, okay, but let's pause for here for just a second. There's a lot that's not shown right now, but what we have here is a cytoskeleton component, a protein, another protein here carrying a vesicle, right, a bag of stuff that's also littered with protein and carbohydrate markers on its surface. Okay, but it looks like from this view, it looks like there's a plenty of open space, that there's not much floating around right next to this vesicle and this filament, right? It looks pretty open. And that's one of the things I want to address is that, while yes, this is a realistic depiction of what it looks like for this molecule to be moving along, carrying this vesicle, right, is life inside of a cell actually is quite crowded, and there's some reasons for that. So I'll post this video and an explanation of this video later, but let's dive in to talk about more about why are cells so small. So again, the average size of a eukaryotic cell is anywhere from like 10 to 100 micrometers. Average for most animal cells is going to be about 50 to 60 micrometers. Okay, so most of our cells we cannot see with the naked eye because we can only see really down to the naked eye, kind of like in this region. We might be able to see a human egg with the naked eye, barely. But we're best kind of seeing at the like 0.5 to 0.1 millimeter scale. Okay, so most of our cells we can't see with our naked eye. They're very, very, very small. Most of the organelles inside of a cell are going to be about the size of a bacteria. So about 1 micrometer for like a mitochondria, maybe a little bigger for something like the central vacuole in a plant, but about 1 micrometer for each of the organelles, the big organelles, right? Why are they so small though, right? Why are cells so tiny? There's got to be a reason for that, right? Natural selection would have selected against any cells that were really, really way too big or any cells that are way, 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 way too small, okay? So the size and structure of a cell has been acted on by natural selection. And what that is, why it's advantageous to be so small, one of the main components of that is that when you're small, you have a huge surface area to move stuff in and out compared to your internal volume, okay? And so what I mean by that is that cells, when they're relatively small, they have a big membrane area, which maximizing that ratio of how much membrane is available, that surface area of the cell that's in contact with the external environment and other cells, having a huge surface area like that means that you can move a lot of material in and out of your cell. And you can be in contact with a large range of different things in your external environment, right? So this diagram sometimes trips students up, and that's okay. But remember, the surface area is just if you were to add up all the area of this side of the cube and this side of the cube and all 6 sides of the cube, okay, the surface area of that cube, okay, would be 6 centimeters squared if it's 1 centimeter on each side. So 1 centimeter times 1 centimeter, this is 1 centimeter squared for this surface of the cube, times 6 gives us 6 centimeters squared. Okay, so that's our overall surface area. That's the amount of the cube that's in contact with the outside world. The internal volume of this cube, right, if this is 1 centimeter on each side, it's going to be 1 centimeter cubed, right? Length times width times height gives us volume. So that means that we have 6 units of surface area for every 1 unit of volume. So we have a huge amount of surface area per volume in this one tiny small cube. If that cell, if we're pretending this is a cell, if that cell were to double its length and width and height to 2 centimeters, we then half our surface area to volume ratio. So like if we had a cell that was 2 centimeters by 2 centimeters by 2 centimeters, our surface area now is 24 centimeters squared, right? We're increasing our surface area, but now if we're 2 by 2 by 2, our internal volume is 8 centimeters cubed. So as we increase the size of a cell, we decrease the ratio of its surface area to volume. So now this cell, if it's 2 by 2, only has a 3:1 ratio of surface area, membrane that's in contact with the outside world, compared to the inside volume of that cell. So it means it's not as efficient at moving materials in and out of its cell, or having a large amount of surface area to contact other cells, or maybe anchor itself, right? So it's not as efficient to move materials in and out of the cell because it doesn't have as much surface area compared to its internal volume. Okay, so that's the key. Being small means you have a huge surface area to move out anything you need to get rid of, to bring in anything you need, food, water, nutrients, molecules, and to be in contact with other cells. So that's the first big idea of why cells are small. Large surface area. The other reason we think cells are really small is because the smaller you are, the faster things can move, right? You don't have to get as far to find an enzyme or to find the substrate or to find the molecule that you're looking for, right? I say looking for as if they have brains, but that's not how it works at all. But the smaller you are, the less distance molecules have to travel to find another molecule for them to react with, which means things can happen very, very quickly. Chemical reactions inside cells can happen very, very quickly if you don't have to go very far. Okay, let's just get a sense of how fast these things happen, okay, and what it's actually like inside of a cell. So remember that image, that video we were just watching, and see how it looks relatively empty in here, right? Well, we wouldn't be able to see any of these components if this were realistically drawn with how chock full of molecules and enzymes and proteins a cell is. Inside of a cell, cells are more like this wave pool that's full of people, okay? They're chock full of molecules, glucose, starch, carbohydrates, proteins, enzymes, RNA, like they are like super, super like chock full of stuff, right? So even though in diagrams we show them as, oh, all these pieces are nice, they have lots of room, they're just floating around in this empty space, that is not empty space at all, okay? There are so many proteins and enzymes and just all of these molecules dissolved in the cytoplasm that are just so close to each other, okay? And so one being small means that things are moving quickly and they're really, really packed very, very tightly together. I am not kidding you. This is how tightly packed it would be inside of a cell. If all of these people were individual molecules, this would basically be what it looks like, but sped up. Okay. So if we were to take one of these people, like if we find a small child, for instance, if that small child was moving at around 250 miles an hour, that would be what it's like inside of a cell. It's not, we're not scaling it. That's just how quickly stuff moves, right? So glucose that's dissolved in that cytoplasm, it's bumping around and moving around about 250 miles an hour, right? Large proteins, big enzymes or big protein molecules are moving around 20 miles an hour. Again, that's not scaled. That's the actual speed they're moving inside of our cells. The difference is that they're not going very far, right? And they're relatively small, so we don't feel our proteins and our glucose molecules zooming around inside of us because they're moving very, 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 very tiny distances, like a micrometer, okay, if that. But what that means, these two things combined together, being super, super packed tightly together like this and moving really, really quickly, means that you can't go very far without colliding into somebody else, right? Like all of these people are all constantly bumping into each other. That's what's happening with the molecules inside of our cells. Because they're moving so quickly, you're going to quickly bump into something else and bounce off, bump into something else and bounce off, right? We are so chock-full and everything is moving so quickly inside of our cells that one molecule, if we were to follow one molecule of like glucose or fructose or something, right? It would bump into another molecule or something else inside the cell about 1 billion times per second. 1 billion collisions per second on average. That's, I can't even fathom, right? And that's just one molecule within our cell. And we have trillions of molecules inside of our cells. So what that means is that sheer frequency, how quickly these things are moving and then bumping into each other, the frequency, the number of collisions that are occurring per second, per molecule, for all the molecules in our cells, means that just eventually, just due to the high chance that you're going to eventually collide with another molecule in just the right way for a chemical reaction to happen. Or collide with an enzyme in just the right way for that substrate to sit into its active site, right? So we talk about oftentimes chemical reactions occurring within cells and like the enzyme and the substrate like to connect to each other, but that connection between these molecules that causes a chemical reaction, it's just a collision that's happening at the right time, okay? And it can happen because there's just so much stuff bumping around and bouncing around inside of our cells, okay? So an enzyme, like let's say we're going to talk about the enzyme, or last week in our proteins lab we talked about the enzyme amylase, right? It would bump into its substrate just randomly, just by chance, because everything is moving so quickly, 500,000 times a second, okay? So things are just moving very, 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 very quickly inside of a cell. And it is just a busy, like buzzing almost of, like a hive of bees just constantly buzzing and bumping into each other. So recap, big idea here, cells are small because that maximizes the amount of surface area they have, the amount of membrane area that they have that's in contact with the outside environment, and because things can move very, very, very quickly. Okay, so I hope it didn't give you an existential crisis of just life is just random bumping into molecules that cause chemical reactions, but that's what it is. All the enzymes that are keeping us alive right now, they're just happening to bump into their substrate just because they're moving so quickly and there's just so much stuff in there. Okay, so I hope when you're looking at these diagrams of cells, you're thinking about all those people in that wave pool and thinking about there's really not much empty space in there. Everything's just kind of crammed in there and bouncing around.